I'd like to begin by thanking the organisers for inviting me. I'm very pleased to be here. This has been something that I think has been waiting to happen for a long time, and I hope that this program goes really well. What I just wanted to show you a few examples about today is the applications of metabolomic methodologies in particular to some of the things that we've been hearing about. The work that I'm going to talk about has been done in collaboration between New Zealand and our laboratory in the University of Manchester, which has been designed, specialised in the application of multi-omic technologies to addressing causation of disease, both in models and in humans, and also attempting to identify predictive algorithms to both identify people at high risk, but also to follow progression. So now, how does this work? Being dumb here. Which one? That one? Yeah. All right. Okay. So, in brief, the main methodology that I'm going to report results from today is the gas chromatograph mass spectrometer. Although for metabolomics also we have a series of liquid chromatograph mass spectrometers as well. And on the right hand side there is just an indication of the type of data sets that we generate. And we can do that in various body fluids, but also in the tissues of the animal or the human. And to structure what we're doing, we also do what's called proteomics. So we can measure up to several thousand proteins simultaneously in a sample of about 50 milligrams, 50 microliter samples. So, and that's a representative GC mass spectrometric metabolomic profile. In this case, that's of post-mortem human brain, the entorhinal cortex, and that gives you that's a low resolution look. Typically, we would be seeing 50 to 100 metabolites in a low resolution analysis, and that we can drive that up to about 1,000 if we go higher resolution. And we have typed, we think, every metabolite in various human tissues that we've studied. Okay, so I'm going to give you a couple of examples of things that we've done in recent years that relate to the metabolomics-based characterization of complex metabolic states. This is one that we published a few years ago. Preeclampsia is a common metabolic disturbance that occurs in pregnancy. It's a major problem, and up until recently it was impossible to predict who's going to get it and who isn't. We undertook an extensive metabolomic profiling study. This is in the blood of at-risk individuals in a program called the SCOPE program that was started in New Zealand. And the upper graph there represents our discovery cohort. The yellow dots are individual pregnancies of people that developed preeclampsia, and the blue are ones that didn't, and you can see that there's a high degree of separation there. We then went to a second country and performed a second validation analysis, which is present in the lower graphs. And as you can see, the thing completely bore itself out. The predictor here is based on an algorithm that involves 14, that's 1,4 metabolites. Interestingly, 3 of them at least emanated from microbes and not from the human ome, and so therefore we presume that these data are saying that some sort of microbial disturbance is probably contributing to the risk of preeclampsia here. But in addition to that, this method has passed the criteria required for application as a screening technology and is now undergoing development in the UK. The point being that there are the screening for this disease that goes on, if you can separate people into high and low risk, which you can here, then this should result in substantive improvements in efficiency. So that's one example of, that was the analysis was done in our laboratory. Here's another one that we completed quite recently, which basically shows that we can do the same thing with prediction of risk of type 2 diabetes. And as Sully was talking about, the risk is conveyed not by glucose, but this is in the time before glucose becomes elevated. We're able to identify a panel of lipids that circulate in the blood that we're able to basically predict who didn't get diabetes. This was done in the UK, so these are Caucasians and South Asians mainly. Doesn't mean that we get the same answer if we do it in New Zealand, but I suspect that there'll be substantive overlap. But what it does show is that the aspiration that Sully mentioned is definitely attainable and should be attainable quite quickly, given the experience that we've had here in doing this type of thing. So that's just 2 examples of prediction of risk of metabolic disease from multi-omic analysis of the blood. So I just want to go on now, and show you a few more things. Yeah. So one of the interests that we've had for quite some time is that of cognitive health. And in particular, how to understand cognitive health. One way or one approach that we've followed, and we've been doing this for about 15 years now, is to actually look at the issue of what causes dementia, because dementia is sort of the ultimate end of lack of cognitive health. And we figured that if we could find markers that perhaps told us what was happening, then we could track back from them. And so this is what I want to show you. Over the last year or so, we think that, and I'm virtually certain about this actually, that we've discovered defects in common, very well-known metabolic pathways. This is within the human brain that are the probable cause of dementia. We were doing it basically in 2 disorders. One of them is Alzheimer's disease, which is the commonest cause of dementia, and the second one is Huntington's disease. The reason we're able to do this is because we've basically learned how to apply these high-power metabolomic analyses based over the top of proteomic analyses as well, that targeted us initially in order to basically elucidate the causation of dementia. Right. Okay, so we published this about 15 years ago now. Basically, this was our initial sort of sighting experiment. It was a proteomic analysis of the brain in Alzheimer's disease. It's the first major proteomic analysis of the brain in Alzheimer's disease published in the literature. And you'll note that at the time, basically we saw a series of disorders in lipid transport, glycolysis, and also a series of proteins that had reasonably well-defined roles in the pathogenesis of diabetes. So this was a study of the human brain, not animal models, human brain. Some years later, we did a second analysis, this time in Huntington's disease. Huntington's disease is quite easy to work on in some regards because the genetic basis of it tells you absolutely about prior risk. And so if you have metabolites linked to, or linked in Huntington's disease, the picture's quite clear. So what we had to do, once again, we identified a link between dementia and diabetes, and what we wanted to do then was to work out what was that and what might it tell us. So this is a study. I'm going to talk about Huntington's first, and then I'm going to back into Alzheimer's. So we published this a few months ago. Metabolomic analysis of the human brain identifies toxigenic urea levels. So we know that these urea levels present in the brain in Huntington's, this had not been found before because the metabolomic analysis had not been developed. We found urea levels in the brain that were sufficient to be causative of neurodegeneration. And there's quite a lot of experimental evidence behind that. In the bottom right graph there, you can see what the table, you can see the actual levels of urea. That's in 11 different regions, so that's right throughout the brain, essentially, and you can see that there's about 6-fold elevation up to levels that are basically causative of tissue damage. And the second thing, more recently, this is in press at the moment, is that we also, in the putamen, P-U-T, putamen, is the region in Huntington's where the disease begins. It's the most damaged region. And we see there huge elevations in the concentrations of glucose, fructose, and sorbitol. These are not present in the circulation. These are just present within the brain. And of glucose 6-phosphate, indicating that the glycolytic pathway is defective. And we also know from evidence that I'm going to show you in a minute, those levels are sufficient to be causative of neurodegeneration. That is the polyol pathway. Polyol pathway is known to be amenable to nutritional manipulation. And here's just Alzheimer's disease. The table on the left is I truncated it just at the first 2 sets of metabolites. But the point is that at the top there, you'll see right across the Alzheimer's brain, there are very large elevations, again, in fructose, sorbitol, and glucose, 2 levels that we know to be causative of neurodegeneration. And I show on the right-hand side there a paper that we just published last month in which we basically applied these methodologies at 3 levels of the nervous system in the rat. That's the sciatic nerve, the dorsal root ganglion, and the trigeminal ganglion. And we know the pattern of neurodegeneration in those animals. And the levels, I should say, in the nervous tissue that cause the degeneration are the same, essentially, as the levels we've found in Alzheimer's disease. Therefore, I think we have proof that we have found the cause of neurodegeneration in Alzheimer's disease. And once more, I would point out that that abnormality there is in a very well-known pathway. It's deeply implicated in the causation of tissue damage and diabetes. And I believe that this will be amenable to nutritional interventions of the types that we've been discussing here. So in conclusion, metabolomic analysis of plasma can be used to predict onset and monitor progression of complex metabolic disorders. I've shown you examples. Common causes of dementia display toxigenic levels of well-known metabolites in brain regions known to undergo neurodegeneration and cognitive impairment. These recently identified by our group Defects provide the probable cause of neurodegeneration and dementia in Alzheimer's disease and Huntington's. Relatively small delays in onset and progression may convey major benefit at the population level. So if we can take these data and find interventions that work on the pathways that we've been looking at, then I think that it would be relatively, well, I wouldn't say straightforward, but the possibility exists, I think, that we can make major contributions. So the pathways implicated in cognitive impairment by metabolomic analysis are well known to be amenable to nutritional and lifestyle modifications, and I'm talking about urea metabolism, and I'm talking about the polyol pathway, and could serve as targets for focused nutritional interventions aimed at improving cognitive health, and development of nutritional interventions to address these recently discovered mechanisms is encouraged. Thank you very much.